Hello everyone, my name is Yanmin Zhang from Shandong University. In this class, we mainly discuss the role of sonic hedgehog in craniofacial development. First, we talk about a brief overview of neural crest development. As you know, the cellular and molecular mechanisms which govern the formation of the vertebrate head and face are remarkably conserved within the entire animal kingdom. Although much of the skull is comprised from mesoderm-derived bone, the majority of anterior craniofacial tissue, including cartilage, bone, and musculature, are primarily derived from the neural crest cells, NCCS for short. Figure A shows schematic dorsal view of tensomite stage chicken embryo, showing the neural crest green in the vicinity of the midline. The dotted lines delimit the embryonic region represented in cross-section from Figure B to Figure E. Figure B, development of the neural crest begins at the gastrula stage with the specification of the neural plate border at the edges of the neural plate. Figure C, as the neural plate close to form the neural tubes, the neural crest progenitors are specified in the dorsal part of the neural folds. Figure D, after the specification, the neural crest cells undergo EMT. Here, EMT stands for epithelial to mesenchymal transition and delaminate from the neural tube. Figure E, migratory neural crest cells follow stereotypical pathways to diverse destinations, where they will give rise to distinct derivatives. Figure F, the neural crest is a multipotent cell population. This sentence means neural crest cells can give rise to mesenchymal cells, neuronal cells, secretory cells, and pigmented cells. This image reveals that neural crest cells migrate from dorsal neural ectoderm and organize the facial development in vertebrate embryos. Colors highlight the region of the developing and adult face that correspond to different neural crest populations along the posterior axis. This figure shows a schematic section through a vertebrate embryo along an imaginary made sagittal plane at the level of the frontal process. You can find many key signaling pathways, including REN3, BMP4, FGFH, SHH, and so on, involved in craniofacial and pharyngeal morphogenesis. Sonic hedgehog, SHH is for short, is one of the three vertebrate homologs of the Drosophila gene hedgehog, first identified in seminal genetic screens by Professor Christine Nielsen-Wohlhard, work which culminated in her being awarded the Nobel Prize for Physiology or Medicine in 1995. This image reveals expression of SHH in the developing craniofacial skeleton at embryonic day 11.5. The expression domain of the floor plate, FP, extends caudally along entire length of the neural tube. Expression is maintained within the diencephalon DI and telencephalon TE, separated by an exclusion zone at the optic recess, O. Expression is also visible in the epithelium, lining the tongue, T, within tooth germs, TG, along the length of both the maxilla and mandible, as well as within the leading edge of the maxillary palatal shafts, MX. This figure is sagittal. Mutations in SHH lead to defects in midline structures, particularly the face and eyes, and a lack of hemisphere separation of the brain. a disorder termed haloprosencephaly, HPE for short. HPE is a relatively common birth defect of the brain, which often also affect facial features, causing closely spaced eyes, small head size, and sometimes cleft of the lip and the roof of the mouth. This figure shows a reduction in head development in embryos after treatment with anti-SHH antibody. Figure AB. Embryos were collected one day and fetal graft at a fixed magnification. 2 measurements were made on each embryo, from anterior optic vesicle to the midbrain, DC, and from the nose to the hindbrain, AB. Figure C. The measurement demonstrates a significant reduction in the size of the head in both dimensions measured. Figure D. 2 days after injection, there was a population of dramatically smaller embryos, with a reduction in bronchial itch development. Figure E. Significantly more abnormally small embryos were seen after SHH treatment, with no increase in embryo mortality. Thanks for your listening.